Lucas graduated from ETH Zurich in molecular biology, and he got his PhD in Dr. Michael Hangner and Proteomics pioneer Rudy Ebersold, and received his degree in 2009 from the University of Zurich. After that, Lucas joined Biognosys in 2010 as one of its first employees. As CTO, he oversees research as well as software and workflow development. Okay, let's welcome Lucas. Lucas, please.
Thank you very much for the introduction, and thanks a lot for having me. As you can see in the title, I'm going to talk about data and AI-driven DI analysis. I changed the title, as you can see, but if you came for the immune peptides, please stay because this part is still in. So first, I would like to talk a bit in general about DI data analysis and how AI can help in analyzing DI data. And everything that I'm going to talk about refers to bottom-up tandem mass spectrometry, as you are all familiar with. And I think I don't need to introduce data-independent acquisition. In this case, you can see data-independent acquisition as acquired in 2 dimensions, the retention time and the fragment M over Z dimension. You have scans on the MS1 and MS2 level, and you basically have an acquisition without gaps if you compare, for instance, to data-dependent acquisition. So everything I'm saying refers to data-independent acquisition, but some of the concepts I'm going to present, they are also applicable to data-dependent acquisition. And yeah. So how do we in general try to process DI data, and what is our philosophy here? So we would start from the spectra. We would first have an identification process. And here we would aim at identifying as many analytes as possible. And as you probably have heard in the last talk, not all analytes are equal, and the quality of not all analytes are equal. So in the next step, we try to make the most of all of these analytes as possible in terms of quantification. And yeah, some of the analytes are higher quality, have more information content. Some other analytes maybe have less information content, but you can apply algorithms that you make the most out of this after the identification process. And then, of course, there are steps after this. There's post-processing, statistics, data science exploration, and so forth. And you can use machine learning or artificial intelligence in all of these steps. I'm only focusing on the identification and quantification part here in terms of AI. And but first, I would like to start how DI data is actually analyzed, because this will help you to understand the different ways how you can use AI for data analysis. So we are doing mass spectrometry, so we have MS spectra, and on the other side we have a peptide search space. So we have a list of peptides which we consider relevant for the sample we investigate, and we can perform, we can try to match the two, going from the peptide search space to the MS spectra, and this is what you would call targeted or peptide-centric analysis. This was actually very nicely described in a paper from the McCoss group. I listed it below there. It's from Ting et al. 2015. But you can also analyze your data going in a different direction, going from the MS spectra to the peptide search space. This would be the more classical way of analyzing your DDA data, for instance, but you can also apply it to data-independent acquisition data. And it's what would be called untargeted or spectrum-centric way of analyzing your data. And then last but not least, you can also try to deduce your peptide sequence directly from the MS spectra. This is what would be called de novo sequencing. Also here you can use AI, but I will not go into this in my talk here. And after you basically have performed your query, and you get a match between a spectrum and a candidate peptide, this would be kind of your hypothesis, and you can perform scoring and confidence estimation on these hypotheses. I will go a little bit in more detail how the two different ways of analyzing your data work, even though this may be familiar to many of you. So for the spectrum-centric analysis, you would typically start with your experimental MS2 spectra, and you would perform a query to the search space, to the peptide search space, based on an index of your precursor fragment ions. And after you have performed your query and you get a match, you will compare your experimental to a theoretical MS2 spectrum. You will perform some scoring. This could be, for instance, cross-correlation scoring or some probabilistic scoring. And then after that, you can estimate your confidence in this match and so on and so forth. So this would be the spectrum-centric way of analyzing your data. And again, as I said before, you can apply this to DDA data, or you can also apply this to DIA data. Now, if you go the other way around, the peptide-centric way of analyzing your data, a typical first step is that you create a spectral library, and you generate, based on this spectral library, you generate extracted ion currents on MS1 and MS2 level. Then you try to detect what we call peak groups, so groups of extracted ion currents, of peaks in the extracted ion currents, who look like a signal from the peptide of interest. And then you select the best peak group, you can perform scoring, machine learning, and in the end, you can perform FDR estimation. So some of these steps are very similar as I just described. Now, if you look at the typical search space sizes you can get for a human proteome, I tried to depict this here. This is in a logarithmic scale. And on the very left, you can see the size of a typical human spectral library. And then next to this, you can see the search space size of a triplet digest of a human proteome. And then if you go further to the right, there is a semi-triplet digest. There is a typical phospho search space for human. And then I also have unspecific digests, which you would typically use for peptidomics. And there, as you can see, the range of peptide lengths you use have a very big influence on this search space. Now, if you look at these different search spaces, you can see that they span a very, very big range, over 4 orders of magnitude of size. And if you think about what they are typically derived from, so the spectral library is often derived or can be derived experimentally, whereas all the other search spaces, they are derived in silico from a FASTA file. And now in recent years, we have observed a trend going from spectral libraries towards search spaces created from FASTA files in the DIA world. And the reason of this is because making spectral libraries is very complicated, it's very time consuming, it's very costly, and hence there have been big efforts to try and go towards the analysis strategies which can start from FASTA files and therefore correspond to easier workflows. And artificial intelligence can help a lot to deal with these large search spaces, and in some cases even enable the respective analysis strategies. So AI was very, very crucial in this trend, going from the smaller search spaces to the large search spaces. Now, if you think about the query, the way I like to think about this is that you would, in the peptide-centric case, you would have your coordinates of your peptide. This would be, for instance, retention time. It would be collisional cross-section, for instance, or it would also be which fragment ions you particularly observe for this peptide. So you have your coordinates and you can go and look for the peptide in your measurement if you're talking about peptide-centric searching paradigm. So you would zoom further in and you would basically find what you were looking for in your measurement. So this would be the peptide-centric paradigm. If you think about it in the way of the spectrum-centric paradigm, you would start with your measurement. You would look for signals that look interesting. And then with these signals, you would go into an index and you would go and look for which peptide this can correspond to. And in both, for both analysis paradigms, you can use AI to predict the coordinates of your peptide, which again would be, for instance, retention time, collisional cross-section, or fragmentation. And so this is how you can describe how AI helps the processes in terms of the query, which is the very first part. So AI enables to predict coordinates and therefore makes this query process more efficient. But it's not only in the query process, it's also in the scoring where AI is helpful. And so again, so predicting physicochemical properties of peptides can be used in the scoring. And if you look at this from a high level, so you would start with your matches. So this can be peptide spectrum matches. If we talk about data-dependent acquisition, we like to call these peptide data matches if we are talking about DA to have some sort of distinction. And then would be your scoring. There is some machine learning involved here, which I'm going into a little bit more detail here, but a very landmark paper here was the Percolator paper from 2007. And after that, there would again be the estimation of your confidence and FDR and so on. And if we zoom into this a little bit more, so you would start with your peptide data matches, and you would perform scoring with classical, what I call here classical scores. This can be, for instance, the length of the peptide, the number of fragments that are matched, and so on and so forth. But then you can also calculate scores, which I would call deep learning scores here, or AI scores. And for retention time, this can be, for instance, a delta IRT score. For collisional cross-section, this can be a delta CCS score, and so on. For fragmentation, you can use cosine similarity. And with this, you can actually derive a very small training data set, and you can perform transfer learning on the models. Why would you do this? This is because sometimes you can see things such as domain shifts in your data. So your models were trained on data that look different from your actual experiment. And in order to deal with this, you can perform what I call transfer learning on the models. You will get improved models for this, or refined models. Again, you can calculate all your classical scores. You can calculate the same scores again with your refined models. And then there is a process, what is called semi-supervised machine learning. Most or all of the modern tools are doing this. It is used to combine the scores into a single discriminant score, and then based on this, you can perform again your ranking, your confidence estimation, FDR estimation, and so on. So AI plays in modern tools plays a very crucial role in the scoring part as well. And again, this can be done no matter whether it's peptide-centric or spectrum-centric way of analyzing your data. Now, to give you a little bit a better feeling for how this looks like. So here I just look into the retention time and collisional cross-section dimension. On the left, you can again see a visualization how you can imagine how this looks like if you're talking about targeted analysis. And on the right, you can see what state-of-the-art predictions look like on a test set for retention time prediction and collisional cross-section. And I show there the relative mean absolute errors. So for retention time, this can be below 1%. For collisional cross-section, it's a little bit higher. It's maybe in the range of 2%. So this is how accurate you can imagine these predictions are these days. And this has been absolutely enabled by deep learning. Before deep learning and these very powerful neural nets were there, this was not possible. There were predictions, but they were never as good as what I show here on this slide. Now, here I show how this has evolved over 6 years. So it's not exactly up to today. The latest data point is around 2 years ago. And I compare here the analysis going from a FASTA file. So this would be the red curve, what I call direct TI here, or starting with, or doing the analysis with a spectral library, which would be a gray curve here. And you can see especially that the analysis using a FASTA file has improved dramatically over these 6 years that are depicted here. And essentially the number of precursors that can be identified from the same data set has more than doubled over the period of 6 years that I'm showing this slide here. So there was a dramatic improvement, which was to a large extent enabled through artificial intelligence. And actually the progression in the last 2 years has gone on in a similar fashion. Now I want to go a little bit more into these models we are using for predicting these physicochemical properties, and especially also into the relation of the training data with the performance of those models. And for this, we will first have a quick look into a different domain, so the domain of large language models and natural language processing. And I cite below 2 papers. One is from OpenAI and one is from DeepMind. And in these 2 papers, they basically looked into how these models scale with an increase in compute and an increase in training data. So you can imagine that for a certain budget of compute, you are confronted with a multi-parameter optimization problem, and some important parameters within there are how many parameters should the model have and how much training data should you use. And in these papers, they basically performed this hyperparameter optimization for a certain budget of compute, and then looked how well the model is performing, or how well these models are performing. And what they could observe is that when increasing the compute exponentially, they could see a linear increase in the performance of those models. And the same also holds true if you look at it from the angle of the amount of training data. So if you increase your amount of training data exponentially, you can see a linear increase of the performance of those models. And what was very astonishing is that these scaling laws, they held true for some of them for over 7 orders of magnitude. So this is very amazing that just by increasing the compute and the training data, and of course choosing very smartly what kind of model you are using, they could see a linear increase in the performance without an end in sight, at least at the time when these papers were published a couple of years ago. And we were interested in how this looks like for models used in proteomics. So this is what you can see depicted here. So here again, we increased the amount of training data exponentially, and we looked at the model performance, and we found that there is more or less a linear relationship between the two, which is of course great news, because it means we can, by increasing the amount of training data, we can further increase the performance of those models. Now you can also look at this from a slightly different angle. You can ask, are the predictions already in a range of the experimental variation, or are we experimentally limited by making use of our prediction accuracy? And so you can basically compare prediction accuracy of your models with the experimental variation, and to simulate experimental variation across experiments. This would be model performance without transfer learning, so not refining a model. To simulate experimental variation within an experiment, this would be model performance with transfer learning. And here you can see the results for the collisional cross-section. And the interesting boxes to compare are basically the turquoise and red boxes, because this corresponds to the prediction accuracy without transfer learning and the inter-experimental variation. And here you can see that these two are actually in a similar range. So I will try to use my pointer here. So these two, they are already in a similar range. So here you could say without transfer learning, you could not further improve or not further make use of better predictions for collision cross-section. Whereas if you look at it with transfer learning, you can see that there's still room for improvement to make use of the predictions. For retention times, it looks slightly different. So there, even without transfer learning, there is still some room for improvement. And if you use transfer learning, then the room for improvement in relative terms is even bigger, is maybe 50% or so. So this means that, for if we are not performing transfer learning, we are already at the limit, more or less, with collision cross-section. But if you use transfer learning, there's still room for improvement. And for retention time, there is still very big room for improvement if we are able to further increase the accuracy of our predictions. And so we basically learned from this, and we increased the amount of training data we use for our models, going from spectrum of 18 to 19. Here you can see a graphical visualization of this. And I will show you a little bit more detail for how we trained our collisional cross-section model. So the spectrum of 18, we used roughly 5 million data points. We trained it with around 5 PTMs. Whereas for spectrum of 19, we increased training data more than, or around 5-fold. We had more PTMs in there. PTMs are important for collisional cross-section because if they are bulky, they can influence your collisional cross-section quite dramatically. So this is why we were interested in including more PTMs. And what you can also see is that we improved data curation. So there are slightly less artifacts in our data. And taken all together, this led to the following improvements. So in blue, this would be the old model. In orange, this would be the new model. What is interesting to see is what I said before. These modifications here, they are basically sorted by their size. And you can see that the improvements are bigger, the bigger the modifications get, which is also what you would expect. So here on the right, the improvements are bigger because these modifications are bulkier. And on the left, the improvements are slightly smaller because the modifications are less bulky. So all in all, we could improve our performance quite very significantly, more than 50%, which then, of course, also has influence further downstream in the identification process in the software. And now a little bit of an outlook. So I want to connect on what I have just shown and talk a bit about immunopeptidomics. So how do our models perform? They are mostly trained on tryptic peptides. How do they perform for non-tryptic peptides? And the relevant workflows here would be immunopeptidomics, probably the most relevant workflow, but in general, peptidomics, or if you perform limited proteolysis, for all of these workflows, it's important to be able to predict also well for non-tryptic peptides. Now, immunopeptides come in two classes, class 1 and class 2. The class 1 immunopeptides correspond to antigens from within the cells. They are presented on the surface of all cells, whereas class 2 correspond to antigens from out of the cells, and they are only presented on certain immune cells. Depending on what you are interested in, you will either look rather at class 1 or rather at class 2 immune peptides. And here you can essentially see retention time prediction in spectrum of 19 on a set of tryptic peptides and on a set of non-tryptic peptides. And you can see that we perform 50% worse for the non-tryptic peptides, which is kind of what you would expect. There are not a lot of non-tryptic peptides in the training data. So we perform significantly worse for these. And so the next question was, can we further increase the prediction of non-tryptic peptides by the scaling laws I have shown before? So essentially, we took two of our models, DeepIRT and DeepFrag, and increased the training data exponentially. And what we saw is what I have shown before, that the prediction performance more or less increases linearly with an exponential increase in training data. So this was very nice to see. The next question we asked was, if we increase the training amount of non-tryptic peptides, does this in some way influence the ability to predict, to make predictions for tryptic peptides? And you could imagine that this is going in both directions. It could go, for instance, towards that predictions for tryptic peptides suddenly are doing worse. But it could also go in the other direction, that the predictions for non-tryptic peptides, for tryptic peptides are getting better if you see more, if the model just sees more training data. And this is what we looked at here. So we also looked at the performance on the tryptic peptides, which are the gray bars here essentially. And what we observed is that it's neither of the two. The predictions of the tryptic peptides just stays flat, which is great. So we don't see a decrease or a deterioration of the prediction of those peptides. Next, we asked how well the prediction performs for immune peptides. Now, immune peptides have slightly specific characteristics. They have certain length distribution, they have certain end amino acids. So it makes sense to look at how the model performs on immunopeptides specifically. And this is what you can see depicted here for many different data sets. For class 1 and class 2, again, for retention time prediction and fragmentation prediction. And what you can see is that the prediction on the immune peptides also improves when going from 0 non-tryptic training data to 41,000 data points of non-tryptic training data. So that's very good to see. And you can also look at this summarized for class 1 and class 2, or all of these experiments I have just shown. You can see that class 1 and class 2 do not behave exactly the same, because they have, as I said before, they have different characteristics, but both of them show a linear trend. And some of this is probably also experimental variation, what you can see here. So we can see improvements for both classes of immunopeptides. And next, we also asked, how does this compare to the non-tryptic peptides, which are also in this case not immunopeptides? And again, you can see that they behave similarly, not exactly the same for retention time and fragmentation. But again, part of this is probably experimental variation, and by and large, they behave rather similarly, which is good to see. So this means that using non-tryptic peptides is probably a very good proxy to train our models for immune peptides as well. So this is a lot of the work we are doing at the moment. So we are heavily working on further improving the analysis of immune peptides. Our models did already see a certain amount of non-tryptic peptides, so they are performing decently, but there is further room for improvement, as I have shown before. Now I would like, in the last section, I would like to talk about data and AI-driven quantification. So of course, identification is one part, but more often people are really interested in quantification, especially if they are performing data-independent acquisition. So quantification is certainly the more important part here. And you can imagine that you are performing a classical discovery experiment where you perform relative quantification. You maybe have two conditions. You perform your MS acquisition, you analyze your data, from identification and basic quantification, and then you perform some statistics. And usually what people want to do is they want to find differentially abundant proteins. So they're also, I would say this is true in maybe 90% of the cases or 95% of the cases. Sometimes people also want to do other things, and it probably depends whether this is more in research or more on the application side. But I would say by and large, people want to find differentially abundant proteins. And the question is now, how can we optimize our software or how can we further improve it for quantification? It's not as easy as in the identification where you just maximize your number of IDs. Here it's a bit more complicated what you actually optimize for. And also you have the problem of ground truth. So ideally we want to have a controlled experiment where we know the ground truth, and this is what we call controlled quantitative experiments, or short CQEs. You will see this a lot on the coming slides, CQEs. And the typical way how this is performed and has been described in the literature is that you would take several organisms and you would mix them in specific ratios. You may have an organism that is there in a fixed amount, so unchanged, and then other organisms that change from condition to condition. Again, you would perform your analysis, and then you can perform your testing for differential abundance. You can rank your proteins by p-value, and you can go in this list from top to bottom and look at how many truly, also known differentially abundant proteins you have in this list up to a certain point, for instance, up to a 5% error rate. And this is basically how we evaluate these controlled quantitative experiments. Now, the key assumption here, of course, is that these CQE experiments are good representatives of real experiments. And in order to alleviate risk, we work with a diversity of different experimental designs and also different LCM methods, because as you will see later, these can have quite a large influence. Here you can see a graphical depiction of the different kinds of data sets we are using for testing. So we use many internal data sets. We use many published data sets where the different organisms are spiked in different ratios, where there are different number of organisms mixed, where the relative amount of the different species are different, and so on and so forth. And here you can see another way of looking at all these data together. So there are more than 60 data sets in total. As I said, there are different LCMF methods, short gradients, longer gradients. And here you can see the performance of different SpectraNode versions, going from SpectraNode 18 to SpectraNode 19, so essentially 1 year, and one monitor parameter of these different versions for one data set shown on the left, and how this is fluctuating over the different versions of SpectraNode. So as I mentioned before, so it's not as trivial in quantification what you should optimize for, because if people talk about quantification, they sometimes talk about precision, or they talk about accuracy of quantification, and which of the two should you optimize for, or which one of the two is more important. So it's not so trivial to optimize for two parameters at the same time. Now, again, as I said before, the way how we do this is we mostly optimize for finding differentially abundant proteins, so only a single metric. But we also look at other metrics that are connected to this, as I said before, precision and accuracy. And for precision and accuracy, there are again several metrics you can look at. So people often look at median CV. However, median CV has one disadvantage, which is if you improve your software and you get more identifications, typically your median CV gets worse because the additional peptides you identify are the more low abundant peptides. And this is not necessarily the effect you want to get. So we think a better metric to look at is the IDs with the number of CVs with CVs below a certain value X. And this could be, for instance, 20%. Now, the same argument you can make with accuracy. So for accuracy, you can look just at the median absolute fold change error of all identifications you have. But again, if you have more identifications, this metric gets worse. We think a better way of looking at it is if you look at the IDs with a fold change error below X. And again, this could be 10 or 20%. And then if you are particularly interested in outliers, you can also look at the root mean square error. I will actually not go so much into this, but it is also shown on some of the other slides. And of course, also connected to all of this is simply the number of identifications you get, because in principle, at least, the more you identify, the more you potentially can also quantify. So again, the metric we use, or the term I will use on the coming slides, is candidate recovery, and this can be done at different percentages. Usually we look at this at 5% or at 10%. And here you can see some results for a single experiment comparing two SpectraNode versions in gray and in red. And you can see that going from version X to version Y, we were able to increase the number of identifications. The median absolute fold change error was decreasing, and we somehow managed to improve in terms of CVs. So the CVs, actually CVs are also getting worse, so the median CV is going up, as you would expect. However, if we look at these candidate recovery curves between the two versions, so at the number of known differentially abundant proteins in relation to the error rate in this candidate list, we can see that there was an improvement going from version X to version Y at 5% error. So basically, we were able to increase the number of correctly of known differentially abundant proteins at 5% error in this candidate list, and this holds true for almost a complete range of error rates. So in our book, this would be an improvement in quantification going from version X to version Y, even though the median absolute fold change error, or even though median CVs were going up, for instance. Now we can look at this on many different SpectraNote versions. In this case, it's around 100 or in this range. And we can look at the correlation of these different metrics here, average IDs, IDs with CV below 10%, and IDs with fold change error below 10%, in relation to the candidate recovery, so the main metric we want to optimize. And for this data set here, you can see that we see the strongest correlation with IDs and a fold change error below 10%. So this in the following slides, you will see this depicted as heat maps. So if there's a positive correlation, it's blue. If there's a negative correlation, then it's red. So in this case, dark blue means strongest correlation across the different SpectraNote versions. However, if we look at this in a different data set, in a different CQE, you can see that the picture looks, again, rather different. So now suddenly the average IDs correlate strongest with the candidate recovery, and the other metrics, for instance, CVs, it's almost flat. So there's almost no relation of candidate recovery in relation to the CVs. So the correlations look rather different, and you can see that it very strongly depends on the data set and the experimental design, which of these metrics strongly correlate with candidate recovery. And now we can look at all these different metrics for the different data sets. Again, here you would just see two data sets, the two data sets I have shown before. You can see all of those different metrics and the correlation to candidate recovery. And we can do this for all the data sets we had, and it's basically sorted by the average IDs in these different data sets. And you can see that there's quite a lot of variation. So there's quite a big dependency on the data set you look at. And what is interesting to see is if you compare this line here and this line here, so the IDs with a fold change error below 10%, which is our preferred metric, as I have explained before, actually shows a somewhat more homogeneous correlation to the candidate recovery as compared to this metric here, because there are some reds here, and you don't have this in here, which is kind of a confirmation of the intuition that this metric might be better suited than just looking at the median absolute fold change error. And you can see the same here again for median CV. So again, if you look at the IDs with CVs below 10%, you can see that this somewhat more homogeneously correlates with candidate recovery. So there's more blue here as compared to the line above, which means that it has somewhat more stable relation to candidate recovery than just looking at the median CV. And yeah, I just want to highlight a few individual cases on the coming slide. So there can be individual controlled quantitative experiments that behave unlike the rest. So this red point here is an outlier, and you can see that in this one version of the software, there was a very big difference for this just one data set, whereas all the other data sets, the correlation is, the change is actually in the other direction. So this is why you don't necessarily want to optimize your algorithm on just a single data set, because it may be misleading if you just look at one instead of many data sets. So, like I said, a version can perform extremely well on just an individual data set, but overall it's actually a worse version of the software as compared to the other one. You can also have subsets of your CQEs behave different. And here this would be in terms of gradient length. So here all the shorter gradient data sets behave differently as compared to the rest. So the red ones as compared to the gray ones. So it can be connected to a certain type of acquisition method. And that's why it's important to have this variation also in the LCMF methods you have in your benchmark data sets. And now we can compare two different SPECNOT versions, SPECNOT 18 and 19, in terms of candidate recovery. So the most important metric, the metric we are most interested in. So this is the metric we want to optimize. And the other metrics, we more want those other metrics to understand better what's going on and to understand what the algorithm exactly is doing. But if we want to see whether the software actually improved, this is the metric we look at. So the candidate recovery at 5% error. And you can see that going from SPECNOT 18 to 19, on average, we saw a significant improvement. There were some data sets where the performance actually got worse, but on average, the performance increased significantly, and this is what we would like to see. And if you would then zoom in into some of these individual data sets, this is how the curves would look like. So here you would see an improvement going from blue to red, and here you would see a slight decrease going from blue to red, but it's rather minor. Now, in the last part of my presentation, I would like to talk about how you can use AI to improve quantification by removing interferences. So this is a model that is called DeepQuant. So you can imagine you have your extracted ion currents of your fragment ions, and here the green one would be a fragment ion with an interference, with an interfering signal that you would like to get rid of. Now we would have our model, and we could feed in the known correct quantities of our individual fragments. So if in an ideal world, if you would know this, we could then have the model predict a correction factor for these quantities based on this training data. And based on these correction factors, you could correct your quantities and get quantities without interferences. So this is essentially what we did. So we trained the model using some training data where we know the ground truth. And here you can see how DeepQuant performs on just one individual example. So there would be a peptide where there is an interference here in this fragment ion. The model is detecting this, is correcting this. And so essentially this exact ion current is reduced in intensity. And then this would then correspond more to the ground truth in the training data for this specific example shown here. But of course, what you want is that your model is not just doing this for individual examples, but having a positive effect on average and in general on the quantification of your full experiment. And then again, what you can do is we can use our CQE setup to monitor this. This what is shown here, 4 different data sets. So here I show the target recovery at 5% error rate in these 4 different data sets when using DeepQuant or when not using DeepQuant. And you can see that even without this outlier, there is a slight increase of 5% going from blue to red. And here it is shown in slightly more detail for different error rates. For 5, 10, and 20% error rates on these 4 different data sets. So on average, going from blue to red, there's a small increase in or improvement in quantification or in candidate recovery, as I've called it before. And here there is a different way of looking at it. So this is a model that was trained purely on timsTOF HT data. And again, the most interesting line here is this line here. So the candidate recovery at 5%. You can see that in all of those different experiments, there was a positive impact of deep quant. The impact was higher on the precursor level as compared to the protein level, but there was a positive effect for both, for the precursor and the protein level. Now I want to note here, deep quant was improving these results on top of the classical interference correction that was also turned on in these cases here. And with this, I'm already at the end of my presentation within time. That's perfect. So in summary, I hope I could show that AI is very, very important in the analysis of DIA data, and it can be used in many, many different places in the analysis pipeline. Actually, in many, many more places than what I have shown here. There are publications for many, many more applications out there. And then what I find very interesting is that these models scale so well with the amount of training data, so that there's this linear increase with exponential increase of training data, which is really great. That we can improve the prediction for immune peptides just using non-tryptic peptides, also very interesting. I find immunopeptidomics an extremely interesting application of mass spectrometry. And then we can further try to improve quantification using a data-driven approach. So using these different kinds of data sets and looking at the right metrics to try and further improve our software. And then AI also shows big potential to further improve quantification by the removal of interferences and potentially being able to do this in a much more holistic way than traditional algorithms. So I also find this a very interesting topic. And with this, I would like to thank you for your attention. And of course, the whole R&D team at Biognosys and the whole team at Biognosys. And yeah. So I was actually not, I will not be at the panel discussion, I assumed. So I'm not sure whether I should take questions now.
Yeah, if you are not joining the panel discussion, then maybe we could discuss a few questions that's been raised in the Q&A. Maybe the first one is, does Spectral not use the actual DIA data to fine-tune models and then apply the fine-tuned models to analyze the same DIA data?
Yes,
anonymous attendee.
Yeah, so what I was calling transfer learning in my slides, Spectronaut is doing this under the hood automatically.
Yeah, there is another one that asks whether, since DIA quantification relies on the highest scoring signature peptides, which have relatively consistent responses across different proteins, does this mean that there's no need to correct for quantitative bias caused by differences in the number of theoretical peptides amongst different proteins? So in the sense that different proteins would have different number of peptides, whether there's any quantitative bias that needs to be considered.
So this is referring to the roll-up of going from peptides to proteins. And this is, of course, a very complicated process, and there are many different approaches of doing that. Now, I'm not sure I 100% understand the question. So whether there what are the important points there to consider? Or, I mean, in general, if you have a very high abundant protein, you will have many peptides to choose from. There will be some peptides that have very good qualities, and you can maybe mainly use those for quantification. And then if you're talking about a protein that is more low abundant, you may not have so much to choose from, and you may just have to use what you have available. And then, of course, approaches like this interference removal and so on become very important. So if you are still able to improve the quality of those few bad peptides you have, then you can significantly improve the way you quantify proteins.
Thank you very much, Lukas. It's a pity that you will not join us for the panel discussion later. But if I can just post a prelude of a question that we will talk about later on and maybe get your opinion. Say, AI is really widely or actively being applied to proteomics, but as the end user, and many of the end users actually do not have the capabilities to use AI on their own. So what does it actually mean to to these end users? How will it tangibly change the way we do our experiments, the way we do our analysis? What is your view on that?
Okay, that's a very good question. So, I mean, there's of course many applications of AI beyond of what I have shown here. So I think, for instance, just in interpreting your biological results, there is probably a lot you can do with large language models. So just understanding the biology better, understanding the role of your proteins, or doing this quicker maybe also. I think that there are already publications that go in this direction. So a couple of groups already work in this direction, I have heard. And I think there is certainly potential there. Yeah, I think you will still have to design your experiments very carefully. I don't think there will be any AI that will do this for us anytime soon. Or yeah, I think it's maybe better if you really understand what is important in designing an experiment, what Mike has shown in his talk before. So I think that's very important, in making the right controls and so on. So there may be the application of AI is of less importance. And then if I refer to the talk I just had, so this will certainly further go on. So there will be better models, the predictions will get better, and the data will also get better. So I expect that there's still a lot of improvements possible in the coming years with better instrumentation and better data analysis. And I hope that particularly for quantification, we will see further improvements, because it is certainly true that we can further improve our identifications, but at a certain point there will be diminishing returns, because there's going to be a lot of noise, a lot of interference in those signals. And if you're not able to remove those, then there will be, the effect will not be so strong. So I think in terms of quantification, there is a lot of potential still. And there's not so many publications in this direction yet.
Hi.
Thanks very much.
How are you?
Yeah.
Could I ask questions?
Yeah, please do. Please do.
Yeah, yes. Hi, Lucas. Thank you very much for the nice question. I'm Terence Poon, the president of HUPO. So I have a quick question. It's about the prediction of fermentation spectrum for a peptide sequence. Now, according to your presentation, it appears that you build your model based on all those DDA data, right? So you train, yeah. But when you apply your AI model, now you are analyzing those fermentation pattern obtained from DIA. Now we all know that the DIA and DDA is relatively pure because the peptides have been selected, and then you do the fermentation. Now it's much cleaner. However, for DIA, within even a narrow windows, there are several peptides, fermented together, and there are certain gas phase reaction happening. Also, during the fermentation, also you change the fermentation efficiency for individual peptide when a group of peptides fermented together. So my quick question is that when you compare the fermentation patterns that are from DIA and DDA for a particular peptide sequence, do you see a substantial difference, or they are highly similar?
Yeah, that's a very good question, and I really did not go into this in my talk. So you can approach this in two different ways, I would say, or primarily in two different ways. So you can perform transfer learning again. So you can refine your models on the actual DIA data, on the actual experiment. And if you have enough of this data to transfer learn on, then your model should be able to understand the differences. But it's of course rather complicated relationship. So it depends on your method, it depends how the windows are placed, it depends on the collision energies applied then. So these are the main differences. But it is a way forward to do transfer learning and try to adapt the models to that. Another way, of course, would be if you would train your models directly on DIA data. But then what I said before applies, right? If it's not the exact same DIA method, if the window placement is different, if the collision energies are different, then this will not necessarily help. But this you can also do. And you can even train your models on DDA and DIA data directly. You can do this by encoding this into the model or perform what we call indexing. But I have to say our experience here is limited, so we don't know the exact effect of this. We did not investigate this systematically yet. We don't know whether models trained in such a way then perform better with or without transfer learning in the wild. So this we don't know yet, and I'm sure there will be publications coming out looking into this, if there not already are. Yeah.
Yeah, thank you, Lucas. As being a end user of those software, so we definitely won't do the transfer learning by ourselves. We look forward to see those better models from Spectralabs or whatever softwares. Yeah.
Yeah, thanks a lot. Yeah, thanks.